we have the streaming, so I will talk to the microscope. We will not talk too much of scientific topics, but rather about the opportunities for the new scientific topics. The reason for this talk was that we got money for the new equipment. And this is a very unusual beast, and we would like to introduce you to this to nanoGUNE and to the Basque community. The story started a few years ago when we started to collaborate with Nicola Abrescia from bioGUNE, who is a group leader there, and started to think about going in the direction of cryo-focused ion beam machining, which is a super fancy topic now in the community because of uprising cellular cryotomography. And FIB is the only method to make the samples for that. So we made a couple of attempts, or let's say more than couple. Here at nanoGUNE developed a special accessories to make it, but our microscopes are definitely not for the cryo work. So when the opportunity appeared, there was opened a call from the Ministry of Science. We applied for this call for the combined microscope for material science and for cryo work. But when doing so, we realized that it's potentially a unique opportunity for the Basque country to acquire the equipment of this level or with these capabilities, and we started to think how we can extend the capacity of this tool to serve more to the community. And the next obvious candidate was nanoGUNE with the batteries, and the thing which we needed to fulfill their request is, let's call it a high rate removal option, which would mean we should have super strong focused ion beam. And it happened that a few, let's say, decade ago, such technology appeared on the market. This is so-called plasma FIB. So on top of the cryo option, it came plasma FIB. And then when speaking about batteries, you are speaking about lithium. And lithium is something which you cannot image in electron or cannot detect in electron microscope because X-ray spectroscopy doesn't go so low in the numbers. And the only way to do it is via secondary ion mass spectroscopy. But the budget was too low, so we asked for the possibility of upgrading this tool for the SIMS spectrometer. And it appeared that the company managed to fit it to the budget. So on top of the cryo high rate removal tool, we got also the TOF, or going to get the TOF SIMS on the tool, which would make it the second tool of this kind worldwide, and the first in Europe. So we will have two speakers today. Evgenii Modin will give the overview of the capabilities of the tool and the research lines which will be opened by the new capabilities. And I asked Nicola to present his work or the state of the art in the cryotomography for you to get the idea about what capabilities we will open in the Basque country for when this instrument will start to work. I would like to introduce you the speakers. So Evgenii got his PhD from Far Eastern Federal University in 2016. Then he moved to Moscow to Kurchatov Institute, where he worked in the group of electron microscopy, in particular dealing with cryo. And in 2018, he joined nanoGUNE as electron microscopy specialist. Nicola got his PhD in 2001 from Universitat Politècnica de Catalunya, if I'm correct.
No. That was the degree.
Ah, okay. No, PhD.
The PhD, okay,
Then he moved to University of Oxford, where he worked on X-ray crystallography of the viruses, to say it in general. And in 2008, he joined Ikerbasque as research professor, and now is leading the Abrescia lab, which is dealing with viral pathogenesis. I'm sorry, I'm reading because this is a new topic for me. Virus cell recognition mechanism and assembly principles of viruses. And I just wonder, it's a great question of Chema, why cannot we have a kind of Pascal lab or Emilio Artacho lab in the nanoGUNE? Okay, so with this I give the word to Evgenii.
So, hi. I'm glad to introduce our new equipment, which will be installed in nanoGUNE during this year. But first of all, let me explain what is FIB and how it can be used. This one is our old system. Yeah. Okay. This is our old system, which we are using during last, I don't know, more than 10 years. And FIB means focused ion beam. This technology is implemented to scanning electron microscope. And here we have electron column, which used for making high resolution images, and liquid ion metal based gallium column. So this column basically we are using for precise cutting of different materials, for TM sample preparation, fabrication of different nano or microstructures, and so on. Also, we have some analytical tools on this machine. They are EDX detector, energy dispersive X-ray detector, which is used for measuring of local chemical composition or building these maps of distribution of chemical elements, and electron backscatter diffraction camera, which we are using for measurement of some crystallographic information about our sample. This microscope has a very big chamber, so additional equipment for mechanical testing or electrical measurements can be installed, and experiments can be performed in situ under the electron beam. And as this machine is a dual beam, of course we can do some three-dimensional characterization of the sample, so we can look not only on the surface but also to look inside the sample. And buying this new tool, the idea was to keep all the functionalities of the previous machine and add some absolutely new possibilities. Let's say combination of possibilities. And these new methods are xenon plasma flip, a secondary ion mass spectrometer, and cryo tool, setup. As you can see, they can be applied to in different fields, as well as in material science and life science. And here at the bottom, in highlighted area, I marked the fields where we can enter only having all this equipment. And they are energy storage and lithium batteries, biomedical application, 3D chemical characterization and depth profiling, sample preparation for cryotomography, isotopic analysis, large volume removal, and gallium-free sample preparation. Let's stop in more details on these methods. And first one is the plasma FIB technology. And here let's compare these two techniques. I mean the old technique which involves gallium as an ion beam, and new technique which uses xenon plasma. Let's compare them in terms of high volume removal rates. To remove a large volume of material, we need a high current in ion beam. And this technology, which works with the liquid gallium, here is the emitter shown. It's a reservoir with gallium, heating coils, a tungsten tip, and extractor electrode. So liquid gallium travels down to the tungsten tip and forms so-called Taylor cone, and emission occurs from this area. But the thing is that this area can survive only in specific range of parameters. So we cannot increase the current as much as we want to get the high removal rate, because this spot emission just emission will stop, and there is no beam. We have this problem in this technology because the emission generates from the cloud of plasma, and a current can be as high as 5 microamps. And in old technology, just like maximum, it could be like 100 nanoamps. So you can see the difference in the currents. The only one thing, if you look at the graph, you can mention that in case of gallium technology, the beam can be focused slightly better at low currents. But after, let's say, 10 nanoamps, it absolutely loses all the benefits, and this technology become more interesting for high volume removal. To illustrate this possibility, we did the experiment during one of the demo sessions on the factory. So this is a lithium-ion battery, and the idea was to open this battery inside the microscope chamber. And so we did this cut by the xenon ion beam. This is a piece of silicon. Here it's shown like a blue part. Then you can see some packaging materials layer, polymer, aluminum foil, and polymer again. And here in the deep, there are some parts of battery. After carefully polishing of this cross-section, we can get this SEM image and check this scale bar. It's 100 micrometer long. And if you look here, this tiny small one. So this is a typical scale where we are working now with the old gallium technology. So you can imagine the scale which we can enter now. What else can we get from using of xenon plasma? Here at this image, we can see the Monte Carlo simulation of interaction of ion beam with the surface of high entropy alloy. As you can see, the interaction volume of xenon beam is maybe twice less than in case of gallium beam. So low interaction volume means low damage to the sample, low implantation depths. Also, xenon is an inert gas, so it do not change the electrical properties of your sample, because it can be critical for some fabrication and further measurement. Also, xenon do not produce intermetallic compounds, and gallium can do this. What is also interesting about the using of ion beam is an analytical application. For example, this is interaction volume of electron beam, interaction volume with your sample. So it's a micrometer large. And in case of gallium or xenon ions, we have a really small interaction volume, so it's beneficial to use the ion beams for depth profiling. This is an example of TM sample preparation, and here we can compare gallium beam and xenon beam prepared samples, and you can see the damaged, so this is the TM images, and you can see the damaged amorphized region is 22 nanometers for gallium beam, and in case of xenon, it's 40% less. The same story we can see with a lower energy for sample prep. So, and this is very critical for transmission electron microscopy, because this amorphized region significantly change the contrast in transmission electron microscopy. So what else? Gallium-free sample preparation. Here we can see aluminum cross-section of aluminum sample and maps of elemental composition. So in case of using gallium beam, we can see that gallium interacts with something on the grain boundaries. So it's changed locally chemical composition on grain boundaries, and if we are studying this alloy, especially grain boundaries, we are already studying some product of chemical reaction of gallium and your sample. In case of xenon, there is no this problem. So xenon is inert gas. It do not produce these intermetallic compounds, and the structure is, let's say, in native state. And this is an illustration where we can see in situ TM experiments, mechanical testing of thin samples. They have been stretched, and these two stress strain curves, they correspond to gallium prepared sample and sample which were prepared with the xenon beam. So you can see that mechanical properties are different. In case of a gallium beam, sample is hard but brittle, and in this case it remains all the plasticity. So it can be really critical if we are studying mechanical properties of our sample. And now let's switch to the next topic. It's secondary ion mass spectroscopy. It's the second tool which will be installed on the new microscope. This is a technique used to analyze the composition of the surfaces by sputtering these surfaces with a primary ion beam, and then analyzing of sputtered ions, secondary ions. And time of flight is a type of analyzer where separation of the ions is based on the time of flight. So it's designed this way that lighter ions, they reach the detector faster than heavy one. What's the benefits of this technology? So with this technology, we can detect light elements starting from hydrogen. Hydrogen, lithium, boron, and so on. Then it's so sensitive that we can detect a trace amount of elements, like ppm parts. Also small molecules can be detected. And it's so sensitive that we can separate different isotopes. Here shown the application of this technology for lithium-ion batteries. This is a cross-section of the batteries, battery cathode, and here shown a mapping of elemental composition. So this is a lithium map for fully discharged battery and lithium map for fully charged battery. So we can clearly see the distribution of lithium inside this structure and how it's changed during the cycling of the battery. What else we can do with this technology? So we can do three-dimensional chemical characterization of our sample. How it works? This primary ion beam, it sputters the surface layer by layer. In each pixel we have mass spectra, and from one layer we can reconstruct the two-dimensional distribution of chemical elements, in this case chlorine. Then doing it layer by layer, we are combining this layer together, getting three-dimensional distribution of our elements. And you can imagine how it can be applied, for example, for thin film analysis. For example, we can do a profiling of changing of chemical concentration across the thin film. And this one is example of this application. So special structure were prepared, where a gallium-rich layer and indium-rich layer following one by one. And here we can see the reconstruction of chemical profile of the structure. But what is interesting, if you look here, I don't know if you can see from there, so the thickness of the layers is less than 3 nanometers. And we can clearly see the changing of chemical concentration across the structure. Then it also can be applied for characterization of two-dimensional materials. Here we can see the molybdenum signal from MOS2 flakes. This technique also can be applied for life science. For example, we can trace the isotopes during the drug delivery. And here is an example, fibroblast cells. And N15 isotope was mapped in the cell, so we can see that the drug is affecting the core. It's really can be interesting for some biomedical application. Also, this technique, TOF SIMS, can be combined with cryo. At this example, and why it's important? Because the cryo allows to keep, let's say, as close as possible to native state of the sample. And in this case, it's kind of some biological object, a tissue or whatever, and this part was mapped to show the different isotope of potassium and how it's possible to bring the cryo possibilities inside the microscope. So we will attach to the microscope this chamber. It's called the sample prep and cryo transfer chamber. And this system uses the cryo stage inside the microscope, cryo stage inside this chamber, so the cold or frozen sample can be transferred through this chamber to the microscope where we have all the analytics and our ion beam. As an example of advantages of cryo technology, we can check these images. So the first one, the top one, it's again lithium related stuff. It's a part of the battery. The top one was prepared at room temperature. The cryo stuff was used for the second one. So, and the cross section were done with the ion beam, and you can see that morphology is a little bit different. At the top image, you can see that probably it's just an artifact, while the bottom image demonstrates a nice and smooth cross section. Line profile of chemical concentration were done for both samples, and you can see for room temperature, probably everything is mixed, so we are studying just an artifact. At the same time, in cryo conditions, we have a nice and sharp interface here. So cryo conditions help to preserve the initial or native structure of our sample. It also can be applied for biological sample analysis. Here is an example of three-dimensional reconstruction of tissue. It's a zebrafish, and some neurons are reconstructed in this bio tissue. Also, cryo setup allows us to prepare cryofracture. It is interesting for some biological application as well as for polymers. Polymer can be frozen, then fractured, and the cross-section can be studied in the scanning electron microscope. Then what is really important about this technique, it can be used for sample preparation for cryo-electron tomography. And if we are lucky to get a TM grid with the cells, we can choose a good cell and cut the cell specific way that produces thin lamella, and then the sample will be transferred back to cryo-electron microscope where some tomographic experiments will be conducted. So, and that's it. The summary, combination of features in this machine is really unique. It's probably the first instrument of this level and possibilities in Europe. We can establish new research lines related to lithium batteries and life sciences. And these extended capabilities can be really served for industry and customers from different sectors, like, for example, metallurgy, where we can measure chemical composition very precisely, or study grain boundaries, or study large-scale defects. Semiconductors and thin film analysis, where we can measure the doping, depth profile, concentration, diffusion, and so on. Polymers, again, we can distinguish different polymers inside the microscope or study chemically modified polymers. Automotive, well, let's say it's metals, polymers, glass, and so on. We can do it. Medical and pharmacology applications, we can work with the frozen cells, with the tissue. We can study drug delivery. Also, we can enter in the field of ecology, geology, paleontology, archaeology, etc. And to be honest, I cannot imagine the sample which we cannot study by using this machine. So if you can imagine something, tell me, it will be interesting to know. And that's it. If you have any questions, you're welcome.
We keep the questions to the end of the presentation.
I might not remember the question.
Okay, first of all, I would like to say thank you to Andrea and Evgenii for inviting me here. I want just to make a point here. The name of my lab, the official name of my lab is Structure and Cell Biology of Virus Lab. The poetic that my directors allow is that I can call it the Abrescia lab. But
that was the question today.
but I don't want creations there. So this is a poetic license, the Abrescia lab. The official is this one. So today my talk in this context is split in two topics. The first one is, I will tell you from where comes from my long interest in electron tomography, and then a project that actually we started very recently as a challenge as well of what Andrei has introduced at the beginning. So in terms of resolution range and system that we study, in my group we study mainly viruses, and we use cryo-electron microscopy, single particle cryo-EM, also X-ray crystallography when we look at some receptor individual proteins. But the focus of today is mainly on cryo-electron tomography or electron tomography. And you can see that the different techniques or the different tools that you can use span from X-ray to PET for looking into humans, for example. So when I moved to the Basque country from Oxford, I was interested in trying to understand the assembly of hepatitis C virus. This was a long time ago now, in 2008. And one of the issue that struck me was the fact that there was no much information about the assembly of this important human pathogen. The only information available in terms of assembly were these 2D images obtained by EPON embedding resin imaging. And this was the schematic pictures of how hepatitis C would assemble. These are the glycoproteins on the ER membrane pinching off. So as a proof of principle that we were able to do some electron tomography at the bioGUNE, we decided to add a third dimension to this 2D view. So we use virus-like particle from hepatitis C using just the structural protein. We put this core E1 and E2 structural protein into a plasmid, and then we overexpressed. We knew that the overexpression of these three proteins originate virus-like particle, and this was a fluorescence imaging marking the E2 production into transfected cells. So this was our first imaging, 2D imaging of control cells. Here the ER is pretty linear. And then we had also cells that were overexpressing these structural proteins. And you can see here these dots, these black dots. These are EPON-embedded cells. This embedding was done in collaboration with the German Andres at the CBMSO in Madrid, and the cutting as well, the sectioning. The visualization was done at the bioGUNE. And these are cryo-immunolabeling marking the E2, and this is a different technique. It's called the Tokoyasu technique method, which marks the E2 at the budding. Now the ER is in white. So once we were sure that we had some morphological change at a cellular level, we decided then to go for tomography. And this is, I mean, most of you hopefully knows that since 2008, there is a JOL 2200 field emission gun at the bioGUNE, which has been recently updated with a K2 camera. But at the time, there was just a 4K by 4K detector. So what we decided to do is collecting data, Tseries data on this microscope. So this is one of the grids that had the section, the cutted cells plastified on top of these grids that then was put onto the tip of this holder, and this holder then enters into the column, and a data collection is schematic, a Tseries data collection is schematically represented by this little movie. So you focus on a region, and then you tilt -60 to +60 at intervals. You collect images, and there is an interval of 2 or 3 degrees depending on what you want to do. And to show you some of the images that we collect on this microscope a long time ago, in 2010, so it's quite a bit of time now. It's to give you an idea of this aligned series is this one here. So this is a plastic. These black dots that you see here are fiducials that allows you to do a better alignment between all the tilted images. Once you have done these, then you do a tomographic reconstruction. Usually we use iMod, which is one of the software that is the best one for performing these 3D reconstruction. Here you can see that this is now a volume. This is a thick volume, and we are scanning through the zeta direction, and you can see this dark. Let's see if I can put it again. You can see here these black dots that they are budding out from the ER in a way that at the time we used was segmenting to visualize this process. We segmented the ER here in yellow, the mitochondria in cyan, and this red bud were the budding particle of this VLP. I don't tell you all the story about this project. This is already all the stories, but we did a sort of morphological characterization of these buds in transfected cells. Now, at the time, we realized that there was quite a bit of limitation in our study. First of all, EPON embedding usually introduce introduce artifact or distortions. Also, first of all, for the plastification, but also for you have to fix the cells, then you have to plastify the cells, and then you have to cut with a microtomo, with a knife, thin sections. And in all this process, there are artifacts that you introduce. Then, at the time, so in 2010, doing data collection. An electrotomographic data collection or a Tseries data collection was not as straightforward as it can appear today. And also, we decided not to go for the high pressure freezing at the time because it's very tedious, at least to me, it looked very tedious and long, but not so straightforward. So at the time, we decided to put in standby the aspiration of pushing forward with the electron tomography, cryo-electron tomography for better times. And when those better times arrived in 2014, 2015, when the resolution revolution in the field of cryo-electron microscopy emerged, thanks to the new detector cameras, for example, the direct detection cameras, through new software for analyzing and for processing EM particles, but also for better microscopes. And at the same time, thanks also to the democratization of cryo-EM, nowadays cryo-EM is not an elite technique. Not only the people that possess the microscope can do or can use electron microscopy, but is much more accessible thanks to many facilities in Europe and around the world. So in 2019, we decided to study the correlation between time of infection and virus-induced cytopathic effect on cellular compartments. So we have some cell, we take virus at different, virus at different MOI, and we infect the cells, and then we take the cells at different time points. And we wanted to look at what type of damage these virus caused to the different cellular compartments. And this was the original idea. Before contacting with Andrei, what we wanted to make sure is that we were getting infection cells infected. So here we are marking the viral polymerase. Here is our control for our processing of the cells. We mark the mitochondria with the mitotracker, then we do some merging. And this was the first step for us to make sure that apart from seeing the cells dying after 72, 96 hours, we wanted to make sure that we were infected. And usually when we do, or we take the cells for cellular analysis or structural analysis, we always run these experiments in parallel. In this case, the cells are always fixed because we are looking at our fluorescence light microscope at the bioGUNE. So with this in mind, then we decided to look around for CryoFIB expertise, and we joined forces with nanoGUNE, actually with Andrei, and we challenged ourselves asking, can we set up a pipeline for cellular CryoFIB? And this was our question at mid-end of 2019. So my talented student, Juan Diego, who was brave enough to take on board this, his PhD student on this PhD on this topic, we are able now to grow our cells onto grids and infect with viruses these grids. Then Juan Diego, what does, he takes these grids, he plunge freeze on our Leica GP2, which is a plunge freezers, and then we keep in our cryoduer, and we came here quite a few times in the past years with the cryo shipper. And then Andrei and Eugene, what they did, they modified at the beginning their CryoFIB, making a sort of cryo station. They did also a shuttle. The shuttle the transfer shuttle is important because it's the machine that takes from the outside world to the inside inside the CryoFIB our little grids. And this I had to put the picture of myself doing something because otherwise I would not appear anywhere, and to show that I do something sometimes, not most of the time. And actually I failed, and then I asked Eugene or Juan Diego to do it. But I won't put those slides. So with Andrei and Eugene, we did a few attempts of CryoFIB, milling of the cells, and indeed we were able to obtain lamella. But what was also important for us to realize is that all the transferring process was suboptimal, as well as the microscope. So that was also the reason why we came out with the conclusion that yes, we can set up a pipeline for CryoFIB, but maybe we need different specs for carry on with this idea. So this was also one of the reasons why the CryoFIB monster, as Andrei called, came or will come to the nanoGUNE. And at the same time, I think that this is a personal slide in the sense that this is my personal view, is that if this machine comes here, there is a likelihood that there will be synergy between different institutions across the Basque country, but not only, also at national level, international level. Most of you know that very recently was installed the first Titan Cryos microscope in Spain, and this is installed and almost operational at the Basque Resource for Electron Microscopy, which is located at the Biophysica Institute in Leioa. So the idea is that maybe we should keep improving our electron microscopy activities in the Basque region, probably, and I think would be beneficial if also cellular groups, which are not usually working with electron microscopy, can see the potential of these technique for their own studies beyond my structural biology studies. And the idea, my personal idea, would be to build an internationally recognized EM hub in the Basque country and I believe beyond. So with this in mind, we had to continue our project. And so while waiting for the CryoFIB monster, we have to keep going and get some data out. And actually, these are very preliminary. These are lamella that we obtain thanks to our access at Ebik, which is located at Diamond, light source synchrotron in UK. And we have been using a CRYO FIB-SEM with gallium system. And we have obtained different lamellas. Well, this is thanks to the Ebik staff, particularly with James Gilchrist, with whom we have been cutting these lamella. Here you can see these lamella. We had, of course, from control cells and from infected cells. Before taking the lamella into the CRYO's microscope, we need to identify by eye the region of interest. And this is something that actually with Juan Diego has done whilst we were obtaining the, while we were visualizing at low magnification the lamella under the cryos. As you can see, it's not easy to distinguish what cellular features you need to focus on. So these circles are the ROI, the region of interest that will be possibly collected under the cryos microscope. And all these are ice contamination, which is one of the challenges, one of the other challenges in this technique. So once you have identified a region that you would like to collect, then you collect a TIL series, which is here to show you. So this is a TIL series coming from these are all, these seem to be vesicles. These are vesicles. The dimension, this sphere, I'm sorry that it doesn't the dimension of this little sphere here is about 500 angstrom. So once we have aligned the TIL series, then we do a 3D reconstruction. So this is a volume. Let's see if it goes. This is a volume. Is a small region of the volume because the size of these big volumes are so, in terms of 60 giga. So we have to actually focus on a smaller part. And the difficulty now is to try to analyze what are these different elements. So this dimension here is about 500 angstrom. You have other objects on this vesicle, and the dimension of those one match with ribosomes, but this is already too much unwavering at the moment. So the idea is that in these 15 minutes that I've been talking, I omitted many details about the technical stuff for a CryoFIB, CryoT project. But there are several challenges that one has to keep in mind, which are the ice formation along the steps. So each step, when you've vitrified your cells, when you then take your vitrified cells and you put in the shuttle for CryoFIB, you can have ice contamination. That's the reason why the cryo station that Eugene has shown you is pivotal to minimize all these steps. But then you have to make sure that you have the right cell confluency on your grid, because you cannot have 60, 70% confluences on your grid, because otherwise it would be a mess. It's very difficult to know the cell state when you are vitrifying. And also, once you are looking at it, also there are inherent problems with the tomography data collection, where the signal-to-noise ratio is not very high, and it's very difficult to identify in the cell, because the cells to me looks like an ocean, and you have to make sure of what complex you are looking at and of what processes you are looking at. So these are some of the challenges that these techniques, this combined technique between CryoFIB and CryoET brings to you. At the same time, there is other aspect that is that we have to consider, and I think is something that we have to think for the future, is that sometimes it's better to integrate CryoFIB with correlative light microscopy. So it can, the CryoFIB milling can be guided by the light fluorescence microscopy. But apart from these challenges that you have to keep in mind, the take-home message from here is that the whole process is very close to native condition, because you have just your cells growing in your Petri dish, you take your grids and you flash freeze that grid. You don't have to do anything else on your sample. And with the CryoET, nowadays with cryos and the high-end microscope, you can have a very fine ultrastructural characterization of your protein complexes. And I want to just remark and stress that our structural study is just at the beginning. It's not the end story. And I am afraid that Juan Diego has to look at many, many, many more, or we have to look together actually, at many, many control cells and infected cells to do a lot of reconstruction and focusing, for example, on ER sites, which are our, or other cellular compartments like mitochondria, etc. But just this was just to show you that this modus operandi that we have been trying to implement with the nanoGUNE, with Andrea and Eugene, it can be implemented for other topics. I mean, my passion is structural virology and the study of viral pathogenesis using structural techniques. But there are many other interests, and this is just a small gallery of papers in which people has implemented, actually developed. Biomaster is the person that has developed the CryoFIB and CryoET combined. And I think is a great opportunity for our centers and for the Basque scientific system to have this CryoFIB monster here. There are also challenges ahead. For example, this will be the first CryoFIB with plasma to be used on cells. There is a huge expectation, and there are methodological tasks to perform, because nowadays everybody uses gallium, but we have been talking and discussing with Andrea and Eugene. This should not have downside, but just benefits for the cellular side in cryo-electron tomography. And I think that with this, I shut up, and I am happy to take your questions. I want just to say that the first part of my of the plastic embedding section, I want to just say thank you to Dave Stewart, who gave me the CDNA for the hepatitis C when I moved here, to German Andres, with whom we did the EPON embedding and the sectioning of the plastic cells, and also David Gilles Carton, who was as well brave enough to make possible the data collection at the bioGUNE at the time. Nowadays, David is at the BREM and is the technical director of the BREM. And today I want to say thank you to Andrei and Eugene, because it's not an easy task to set up a cellular CryoFIB. At the Durne Berra and Laura Palomo from Caracedo Lab at the bioGUNE, who helped us at the beginning with the fluorescence microscopy. And of course at the Ebik staff, James Gilchrist, who has helped us with the lamella that I show you today. And of course, the people in my lab. A part that I didn't explain you is the fact that actually we are not just using commercial cells, but we are also using primary cells, because we want to see the infection on primary cells. And with this, I close. Okay.
Thank you, Nicola, for coming in for giving the talk. Before we start with the question, I would like to make two notes. First, all the technical stuff, all the complements for the technical development should go to Chris. And I would like to just to mention him in this respect, because all the things which were constructed for cryo are coming from him. And the second thing, You probably are quite concerned about the viral stuff which is going to appear at nanoGUNE. I would like to say that lab 4, where we will install the microscope, will be rebuilt for biosafety level 2, and the microscope will be inside the biosafety lab. So this is similar to what Raoul has on the ground. So now the talks are open to discussion. You forgot.
Can you hear me? Well, I remove it. No, it was a curiosity. Now that you envision that now using, or tomorrow using xenon, you still need to put the platinum before the cut, or you change anything there, you can remove it? No, it's still needed.
Some silicon, if you are cutting something big, or anyway, you have to put the platinum constant. So this is the same.
It's always needed. Okay, thank you.
Thanks, Eugene, for the talk. This is kind of another technical question, because this xenon plasma reminds me a lot of the argon plasma that we use for, ion milling in general. So what are the advantages of having xenon apart from having more weight in the atoms?
Okay. Well, some manufacturers offers like 4 different ion beams in one machine. And these different ions like xenon, argon, nitrogen, oxygen, they can be beneficial for different applications. Like, for example, biological stuff can be better cut with oxygen or whatever, but xenon is like proved that it works well for nearly everything, and we can cut whatever we want. With the argon the same, but in this case we are using xenon. Yeah.
So maybe a bit more physics into that. The xenon is heavier, so at the same energy it's lower, so the damage is less.
Yeah.
But the removal rate efficiency is the same or better, so you get all the benefits. The better would be to go even deeper in weight, but then it will be too expensive.
Right. Thank you.
You're welcome.
Yeah, I have a question actually for Nicola. You mentioned at the very end the transition with the new cryo monster, or what it's called, from gallium to now the iron source. And I was wondering, it seemed from what you said at the very end, there's very little known about this. Is that really the case? People haven't experimented with it, because then it seems like a risky strategy to jump on that technology.
I would better answer that he's very skeptical.
We
We
have had quite a bit discussion. There is very little in literature done on using xenon plasma on cellular, on cells. But there is one nice paper, I didn't actually wrote the reference, in which they comment about the possibility of using xenon plasma on cells, because this would be the next step on all the advantages on material science. I have to say that the CryoFIB for cellular comes from material science. That is very important. So I was skeptic, but as well convinced by Andrei, because material science is the expert, that the transition to cellular sample would be smooth. And I think I will let you.
Yeah, I was also very concerned about this, because as Zhenya told, the focus is significantly worse for xenon, just because of the size of the primary source. That's why we went for demos, and most demos didn't have any problem to make it.
Yes.
Also, it's just related to the amount of systems. The gallium systems are nearly everywhere, and these xenon systems, they're
way more expensive.
They're new and expensive.
So probably.
We're breaking the new ground.
Exactly.
As well. Okay.
Maybe
Maybe it goes south, but at least we're trying something new here.
exactly.
Okay. Maybe just an add-on question, but that's to Jamie. The gallium, you said the gallium has such a nice Gaussian beam, but I remember when we were trying some magnetic multilayers back then, it seemed to have rather substantial tails too, right? So how am I supposed to understand that? Then with the ion source, it gets yet far worse, or maybe people just oversell the precision of the gallium source?
Zhenya has shown the data from the paper, and to be honest, I don't understand the physics behind what was told there, because physically the gallium source is a kind of a point source, and you get the tails because the scattering, the emission goes from the side source. That's why you get Gaussian, because all the optics you focus on, you get Gaussians, and the tails are quite far. In the case of plasma, you have the plasma volume and then the opening. So in principle, if you focus exactly on this opening, you should get a rectangular shape. And when you are milling, important is not the shape, but the edge, so the derivative. So in this sense, xenon or plasma sources should be beneficial. I mean, the guys are telling different, but I really don't see I'm not convinced by this one. So I see that, or I can imagine that xenon is really beneficial in the sense of milling from the side, not from the sense of resolution because the beam is still broad, but from the size of precision of milling, yes.
Thank you.
Okay, so can I have another question, joke, something like? So we have been doing cross sections for us for quite a long time, and they're great, right? But now you will show us how this xenon plasma cryo is going to be amazing. So how are you going to convince us to do things with the old machine? Because now, I mean, when I give you a sample, I would say, no, but I want the good one, right? I don't want the old one because this is so much better. We are going to see things which are expensive. It's going to be so expensive that we cannot pay for it, and then we'll have to go to the old one, or how does it work?
Concerning expenses, it's not the question to us. I can assure you we will always make the best samples for you.
Oh, that sounds so good.
But now...
no, I mean, it's a joke, but it's also a fair question
no,
in the sense that in
is
which samples we will not see the difference, for instance? Because there may be samples that, well, we don't really care to do cryo.
We will not see the difference in any way. We will see the difference because it will be much easier.
Okay, that's a good answer. Because actually we don't understand anything, we just write whatever you tell us.
What I want to say you, we will not study 80% of your samples with cross-section anymore, because the most thing you are asking, we need the depth, penetration depth of interdiffusion between two layers. Mm-hmm. We don't make a section for that. We make the depth profiling with topsums. And then you know it to PPA. And that's much easier and much faster.
I don't really want that precision, it will be terrible. The samples will be wrong.
No, but the depth resolution is not that great.
About 3 nanometers.
Oh, it is
3 nanometers.
I see, okay, I got that wrong.
No, it's, but I mean, the period of the latches was like 5 nanometers.
3.
Yes.
No, 3.
Yeah, 5.
Cool, thanks.
And we are going to work on it, so it will be better.
Correct.
But that was gallium, no?
No,
that was xenon.
Oh, okay.
Can I ask another technical question? So when this will be installed?
We should talk to lawyers. The papers for tender were supplied the 2nd of March, and they are still there.
Very interesting approach.
So we plan to open the tender in the first week of March, but
okay, at some point.
So the aim was to have it to start installation in October and to finish by the end of the year, but
don't know